environmental section, and I'm glad to be here today to introduce you a bit the PhD project I'm working. So I kindly invite you to scan the QR code for the antennas. Can I go on or? Here we are. As you read from the email, this is a provocative title, and I'm actually working on waste valorization in general, together with Professor Elena Ficara and Professor Andrea Turolla, with a co-supervision also from the University of Valladolid. And my project deals with finding values in something that usually is meant as a mere waste. So let's start. Anyone who has told me at least once knows that I'm not used to include bad words in my language. But in this case, I really believe that from sheet to gold is the best expression to describe my PhD project. Jokes aside, this is not only my idea. This is also the idea of my colleagues that share the lab with me, and also the smell of my of my waste. That is why they made this for me before I left for my visiting period. So thank you, Giacomo, for this. Jokes aside, I'm really proud of what I'm doing. And I think one of the main challenges of my PhD project is learning to look beyond appearances. And by learning this challenge, I had the opportunity to see something different, to see value when normal people see waste. To take on this challenge of looking beyond appearances, I need some tools. Between them, the most important one are my glasses. My glasses have a beautiful superpower. Yes, today I'm wearing contact lenses, but actually I can guarantee they have the same superpower. When I wear them on, I have the possibility, the superpower, to see beyond the surface, to see something different, and see it very clearly. Now, today I'm here So today I want to offer you the possibility to wear my glasses just for a while and see what I see, so from a different perspective. Now you may be wondering what kind of materials these glasses are made of. Well, there are three main ingredients that are constituting this pair of glass. The first one is awareness. The second one, curiosity. And the third one, knowledge. As I'm not an optician, I can't provide you with 40 pair of glasses, but today I'm here to let you build your own pair and see clearly the transition between from sheet, that's a bit to gold, of course. It's a bit slow, but let me bring you into the context. Okay. So let me bring you into the context. And it's a scenario of what the world would like if nothing changes. So close your eyes. Imagine we are on planet Earth in 2050. 9.5 billion people are living on the planet. We are more hungry. We need for more energy, more of everything. But the resources are not growing with us. They're shrinking. So imagine crowded cities, exhausted soils, empty shells, no light, silent plants, because there is not enough energy to run them on. According to FAO, 50% of habitable land is exploited now for agriculture. And only 38% of cereals are directly used for human feed. Less than half. The remaining part is addressed to animal feed, without taking into account the amount of waste that are generated every year from the agricultural sector. In the past 60 years, the global protein demand is more than double. And in few countries, it reached 10 times the initial protein demand. And if we zoom in on soy, one of the most abundant crop for feed applications, we can see that 76% of the total produced soy is addressed to only animal feed. Only 1/5 is addressed to human food. So from this data, we can see clearly that there is competition between feed and food sector. The pressure is not only on agricultural sector, but also energy is under pressure. As we can see from this graph, in the past 60 years, the energy demand is enormously increased. This is because our lifestyles, our energy demand is growing more and more. So now we see the whole perspective. All these issues are not separate. They are very interconnected. Increasing population leads to need for more food and feed, but also more energy. And producing more food and energy will lead to more waste and emissions. So we are not facing a single issue, but a very complex problem. So now, what would you say if I told you that a single technology could solve all these problems at once? Of course, it's impossible, right? But I'm working on it. What if we change the perspective and the whole system? What if we can produce feed from waste and emissions while producing sustainable energy? But let's take a step back from where the problem starts. Anaerobic digestion. Anaerobic digestion is a quite consolidated process that is aimed at converting organic waste, in our case, cow manure and crop waste, to valuable biogas, which is a sustainable energy source. This is a good strategy to meet the increasing energy demand. But this process is not perfect, as it still has a lot of byproducts. Anaerobic digestion mainly generates CO2, that is an impurity of biogas, and digestate. Many of us already know very well CO2, which is a gaseous byproduct that generates, well, that contributes to global warming. But many of us maybe don't know what digestate is. Digestate is a semi-liquid byproduct of anaerobic digestion, which is still rich in nutrients, such as nitrogen, phosphorus, and so on. And because of this, it has been used for years on soils as fertilizer. But as the number of anaerobic digesters is increasing in order to meet the energy demand, so does digestate, and so does CO2. That's why several pollution environmental issues arose, such as eutrophication, and the European Union had to strictly regulate its application. That is why the perspective is that the amount of CO2 and digestate is going to increase in the following years. And it's harder and harder to manage them. But what if we can make this technology chain more efficient, more sustainable, by converting these byproducts into protein for animal feed? In this way, we could be able to valorize the nutrients that are in digestate and the value of carbon sources in a bioprocess and combine them to produce something valuable. This way, we could be able to close in the loop of nutrients, but also the carbon cycle, reducing the emissions of this process while going on producing sustainable energy. But let's have a look. This is the focus of my PhD project, because I'm working on a biorefinery based on bacteria use, in which the pre-treated digestate is separated into phases. The liquid phase, which is rich in nutrient, is used as nutrient source for the bacteria. They are in a bioreactor together with the gases. The gases of anaerobic digestion are mainly CO2, as we said before. But then we developed another strategy to valorize also the solid phase of digestate. By gasification, we can obtain a gas mixture that is rich in hydrogen. Hydrogen is because we also need an energy source for this bioprocess. The final product is a biomass, bacterial biomass, rich in protein, that is currently used as protein supplement in animal feed. But why agricultural digestate and not other types of waste or liquid streams? Well, agricultural digestate is very good, as we said before, is rich in nutrients. But compared, for example, to sewage sludge and to food byproducts, it's safer, as the heavy metal content and pathogen is limited compared to the other sources. So it's a good strategy to go on with this. Microbial proteins, in general, are microbial dried cells used as protein supplement in food and also in feed and food applications. There are many ways to produce them in biological ways, so different routes that are exploiting different types of feedstocks. But the research frontiers aim at producing them from waste feedstock. That is why we selected the hydrogen route, which is based on CO2, biogenic CO2, and green hydrogen for their production. So as we said, biogas can be addressed to upgrading, a consolidated process that separates methane from CO2, and biomethane can be addressed to grid, while this CO2 can be valorized as carbon source for our bioprocess. The second route that we explore is gasification of solid digestate to obtain a mixture rich in hydrogen, which is usually expensive. This is a summary of how we passed from an idea to a validated process. We tested 2 different bacterial strains in 2 different gas mixture configurations. The reactor we used were semi-continuous, and the gas have been replaced once they were consumed. So during this experiment, we were able to assess the biomass growth, the protein content, and the gas consumption. We obtained remarkable results. Actually, previous literature were based on protein production from mixed culture. And not only from CO2, but also a variety of substrate, such as organic acids or methane also. But methane, from a preliminary techno-economic evaluation that we did, is not so profitable as it already has its value as energy source. Mixed cultures are preferred because they are easy to manage and to grow. However, they have an important limitation, as once we need to authorize the product on the market, it is very complex and expensive, as every strain present in the mixed culture needs to be identified and declared safe. That is why recent research passed through pure cultures. But as we can see from here, the protein content of the biomass was lower than the protein content obtained in mixed cultures. In our results, we obtained values comparable to mixed culture growing pure strain, pure bacteria, using waste substrates. As we said, with two different gas configurations, hydrogen, CO2, and syngas, but the nutrient source was not a synthetic one, as in all these cases, but it was made of nitrogen and phosphorus and microelements present in digestate. These results highlight how the micronutrient content of digestate could promote the biomass growth and boost the protein accumulation. We also assess the quality of the protein we produce. Compare them with traditional feed, such as fish meal and soybean, but also with the most comparable results found in literature for microbial protein. To assess the quality of protein, we had to evaluate the amino acidic profile of the protein, as the amino acids are the building blocks of protein, and they determine the nutritional value of the final product. As we can see from this graph, we see that the results we obtain, that are the first four columns, blue, light blue, green, and light green, are comparable to the amino acid content of traditional feed. Moreover, all the essential amino acids were present, and this is a promising result, as we can see that this product could be as a good substitute for traditional ones. In two cases, we saw that our values were lower than the values obtained in fish meal and soybean as traditional feed. But these two amino acids in particular, aspartate and glutamate, are responsible for flavor. So a lower content in the final biomass is not an issue as regards the nutritional value. This means that our protein will be a little bit less tasty than traditional feed. And thanks to the good results we obtained, we are now waiting for our pilot plant that will come in the following weeks to scale up this process. Here we have the reactor. 2 weeks ago, we went to the production site to see it. And in this way, we were able to test in continuous mode with a higher scale the results and collect possibly a lot of biomass and protein. So probably we succeed in changing a bit the system, as we demonstrated that it's possible to produce food and feed in a more sustainable way, less reliable on soils, in a more efficient ways in which nutrients are recirculated and emissions are limited. By converting digestate to protein, we are not only producing value from something that is usually considered a waste, but we are also optimizing a more consolidated process, which is anaerobic digestion, as we were able to reduce the amount of byproducts that we address to land. And also, we are limiting the emissions that is generated. So let's go back to our scenario. We are always on planet Earth. Always 2050. The same number of people are living on the Earth, 9.5 billion. The same need for food and energy demand. But something has changed. We learn how to respect limits that our planet is posing to us. We learn how to optimize resources. There is not collapse, but resilience. And if I now go back to the question, what would you say if I told you that a single technology would solve all these problems at once? Maybe your answer has changed a bit. Of course, it's not easy. There's still a lot of work to do, and of course, this won't be the sole technology that could save the world. But still, this is a possible alternative to be explored in order to optimize the processes that we already know. And if I've done my job today, maybe something has changed your perspective. I really hope that now, with your new pair of glasses, you can see a little bit clearly and a little bit closer that the transition from sheep to gold is something possible. Thank you.